Okay. We are on. Okay. We are on. We are on. There we go. I'm going to get this thing moving. Oh, everybody ready for a show? Everybody
born ready.
Yeah, recovering from Halloween. There we go. We are starting in 3, 2. This is TWIS. This Week in Science, episode number 643, recorded on Wednesday, November 1st, 2017. A wonder. I'm Dr. Kiki, and tonight we are going to fill your heads with an electric shock, lasers, and little oysters. But first,
disclaimer, disclaimer, disclaimer. Nobody gets out of here alive. It's not a threat. It's just the cards you've been dealt by being alive, that one day you will shuffle off this mortal coil. And in the meantime, everything you see, hear, smell, taste, and touch, this is the world. And yet, the world is much bigger still than this, for there is knowledge that can take us far beyond see, hear, smell, taste, and touch. Knowledge that can fill our everything with just about anything, from the Big Bang to the microbial muck, from genomic manipulations to rovers on Mars, from basic chemistry to advanced chemistry, and theories on how gravity works. Knowledge is to be found everywhere, and we humans are adept as can be at it. And every bit of new knowledge that we have gives our world, our life, our everything a little more. This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
and good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back once again to exclaim and explain all the wonders of the science world. And I don't know if any of you caught the reference of the title of the show this evening, A Wonder, from The Tempest. November 1st, a long, long time ago, was the first public performance of Shakespeare's Tempest. Which,
I had no idea.
Yeah, which has...
All right, Justin, rewrite the disclaimer. Let's start over.
Let's start over.
I had to put in some sort of Shakespearean reference, like shuffling off a mortal coil. I had to have done that.
That's right. But instead, what's past is prologue. And it's into the show we go.
Oh, yeah.
It's so good. Oh, I love this show. I love our audience. You know, hell is empty and all the devils are here.
Nice.
Okay, but I'll stop.
Maybe at some point in the after show we will though get the that what's that voice you use, Blair, when you do Shakespeare?
Vocal fry.
Vocal fry Shakespeare by Blair. It is worth it.
Okay.
It is worth it.
Maybe.
Okay, let's hold off on the vocal fry until we get
Sorry,
to the later
everyone,
parts.
Don't leave. You can leave later.
The vocal fry comes later. Right now, we're going to talk about science news, and we have tons of it. I have news stories about electrical stimulation for the brain, cooperating chimpanzees, and inevitable aging. Justin, what did you bring?
What do I have? I have salty saliva,
Ew.
primordial soup, alien physiology, and traveling at the speed of gravity.
Ha. Okay, I'm excited about these. I'm excited. Blair, what is in the animal corner tonight?
Oh, I have some marine monsters. I have squeamish shellfish, and I have T-Rex arms.
Makes it hard to get things
Never-
off the top
I
shelf.
can't cite the
Mm.
T-Rex arms.
Never noticed that about you.
I know. Yeah. We need to
Ooh.
spend more time in person to see things like that. No. But anyway, as we jump into the show, I would like to remind everyone out there that you can subscribe to the TWIS podcast, all good places that podcasts are available. iTunes, Google Play, Stitcher, Spreaker, TuneIn, probably others as well. We're on all sorts of places. You can also just look for us This Week in Science on YouTube, on Facebook, and you can visit TWIS.org and go there, especially if you are interested in finding out about our 2018 TWIS Blair's Animal Corner calendar, which is now available for pre-ordering. And also, everyone, if you are in an entomology field and going to Denver, Colorado for the Entomological Society's meeting in Denver, Colorado this week, we will be there. This Week in Science is doing a live show at the ESA, the Entomology 2017 conference in Denver, Colorado. It'll be on Monday, which will be a lot of fun. We're excited, you guys, right?
Yeah, and if you're in
Yeah.
Denver and not an entomologist, we're probably going to do a meetup as well, right?
Yeah, so keep your eyes peeled on the social media account, because when we figure out where we can do something like that, we will tell you, but we don't
Yeah.
know yet. So we'll get
No,
there.
because we're not from Denver.
No, yeah, we don't know these things. All right. Moving into one of my favorite segments of the show. What has science done for me lately? I have an update to last week's entry from Selden McCabe. I didn't see he had sent another note and said, "Hi, Dr. Kiki. Actually, there's more to my story." In 2010, and this Selden last week, he talked about how technology allowed him to share his experience of the eclipse with his friends around the world. He says, though, there's more. In 2010, I was diagnosed with stage 4 cancer with a 28% 5-year survival rate. Thanks to science, new discoveries and treatments have given me and countless other cancer patients more time to spend with family, traveling, and time to enjoy life. Thank you, science, and thank you, TWIS, for reporting on the latest science news. And Selden, thank you for sharing some of your absolutely precious time with us. And we are so glad that you shared your story with us.
Mhm.
It is just wonderful. Then we did have another letter from Minion. Let's see if I can get her name here. Holly Hunter from Oklahoma City. Holly Hunter says, "Dear Dr. Kiki, Justin, and Blair, I live in Oklahoma City where science struggles for respect. But we also have a lot of wildlife out here, and that's where science does so much for me. I am a pollinator gardener. And thanks to countless hardworking entomologists and botanists, I have an X-ray view into the world of the fascinating insects that populate my gardens. Thanks to their studies, I can look at a monarch butterfly and fully appreciate the struggles that she has overcome just to reach my garden. And it is deeply satisfying to know that I am directly contributing to the survival of a threatened species. And I only know that because of scientific research. Scientists allow me to peer into the inner lives of bees. For example, did you know that queen bees determine the sex of their babies by choosing whether or not to add sperm to each egg? Entomologists told me that. Here's another fun story about bees, full of intrigue and internecine warfare. Bumblebees are quite different from honeybees because their queen isn't the only one who lays eggs. In late summer, her daughter worker bees also lay eggs. Isn't that amazing?
I
But
didn't know
wait,
that.
I know, but wait, there's more. The mother bee eats her daughter's eggs, and they eat hers, and the reproduction battle is on. By the end of the season, the colony will end up with a certain percentage of males who are not the queen's sons. Wicked crazy, right?
Yeah. No, I did not know that at all.
No idea. Thank you, scientists, for making this amazing discovery. And thanks to the scientists who are working hard to find out how to save our pollinators. Our lives would be so boring without the bees and butterflies, not to mention their critical role in agriculture. And last but not least, thanks to our science mentors, like you guys. I enjoy your spirited debates. Justin's crazy ideas are sometimes really spot on. I'm so glad he thinks outside the box. And I'm also really glad that Dr. Kiki and Blair are there to challenge his really crazy ideas. There's nothing more true to science than a lively debate. Keep up the great work. Holly Hunter from Oklahoma City. Thank you,
Holly.
Thanks, Holly.
Yeah, Holly, that was a This Week in What Has Science Done for Me Lately that was full of information that we weren't necessarily aware of. And that is so fun. Thanks for educating us in this wonderful letter. And yeah, pollinators. Let's find out more about them and how we can preserve them.
I wonder where we could do that. Oh, wait. How about the entomological conference?
Yes, let's do it. We're going to do that. We're going to find out more for you, Holly. We'll get some good insect news for you about pollinators. Everyone out there, please write in with your stories, your tales of what science does for you, what it has done for you lately. Let's keep filling this part of the show with your emails, with your notes, with your stories, with your lives. It makes all of our worlds a little bit richer and better. Thank you to everyone. But keep it up. Leave us a message on our Facebook page. Facebook.com/thisweekinscience is where you go. And just send me a note and I will schedule it to read it on the show. Let's move on. You guys ready?
Let's do some science.
Yeah, let's do some science. Oh, politics these days, it makes me think that humans will never be able to cooperate again, right? Things get me down. They get me down. But
maybe not here.
Not here on the show. No.
Wait, do we have internal space?
I meant in America, but
In
you go ahead.
In America, yeah, maybe, but there's evidence of cooperation all over the place. And you know what? Even our closest cousins cooperate. There's evidence. Study from Kyoto and Oxford universities and the Indianapolis Zoo looking at chimpanzees. Well, I guess not our closest because bonobos are supposed to be our closest now, but fairly close. They have been taking turns and cooperating to complete a number sequencing task. They're counting in the proper order, or at least they're recognizing symbols in the proper order to receive a reward, of course, but they're coordinating their behavior in a very, very rapid fashion. So this research is published in the journal Scientific Reports, and the researchers trained the chimpanzees in using a digital touchpad with numbers on it. And the chimpanzees had to touch the series of numbers in the correct order. And they had gotten to do it on their own very well. So they could tap the squares on this number pad. And even though the numbers are not in any order, they were arranged randomly on this square, the chimpanzees could see which number came next in the sequence and tap out 1, 2, 3, 4, 5, 6, 7, 8 and get it and finish the sequence. But they'd never been asked to share the task and to have to coordinate their activation and their activity. So in this exercise, what they did is they split the touch screen and they had the chimpanzees so they could see each other, but they were separated by a wall. And the touch screen, one half of it was on each side of this wall, with one chimpanzee having access to each side. Now, the numbers were randomly allocated between the different screens. There we go. I'm gonna get this video big so you guys can watch exactly what happened in this situation and see how this turned out. And it's actually pretty amazing how rapidly these chimpanzees share the task. They spontaneously are able to touch the correct numbers to be able to finish the sequence.
Wow.
And gain a reward. It's
Okay.
it's
So
a fast cooperative process.
But I've seen this lab before too, which is really amazing. Is one of them is a second generation of the lab. His mother was in the lab, and the son chimpanzee's in the lab. And he can do that sequence. I don't know if it goes up to like 14, 15, 16 numbers, where they flash it on the screen for a fraction of a second. Like, you can't even read it.
Yeah.
Your human eye cannot read or place for that instant where all those numbers are, and they can hit those squares in the correct sequence. But this is a new task where you pause. You don't just hit them as quick as you can in the order. You pause to allow your partner to engage the other part. That is fascinating. Wow.
It is fascinating.
I feel like there's a lot of small children that definitely could not do that.
No,
right.
Adults cannot do this.
And so this is
the cooperation one. The one they just did, the one before, though, they were unable to.
And so the interesting aspect of this study is there were 6 chimpanzees, 3 mother and offspring pairs. And so they were all really good at it. And as you said, Justin, the young chimpanzees are really good at it, and they don't make that many mistakes. They made fewer mistakes than the mothers, actually. And they're faster than their mothers. But during the control test, they found that the mothers are faster when they're all by themselves and they're not doing it on their own, not doing it together. And so what it suggests is when they're doing it together, the young chimpanzees are much better at paying attention to what their mothers are doing than the mothers are at paying attention to what their children are doing.
I would think it would be the other way around, but yeah, wow.
Yeah. And so one of the researchers adds the finding that young chimpanzees more readily took cues from their mothers when looking to take their turn reveals interesting parallels with other aspects of information transmission in chimpanzee societies. For example, during the learning of tool use by wild chimpanzees, we also see young individuals paying attention to older ones much more than the reverse. This kind of asymmetry has important applications for the direction of information flow. For example, how quickly new innovations in behavior will spread through a group. So I think it's a fascinating example of cooperation in a social group, a social species as well. But it's a fascinating concept that this kind of coordination, this level of rapid coordination, can take place on a task that really has nothing to do with their natural behaviors. So the implications for what they can do coordinating together as a group in a wild on natural behaviors, I think could be much more interesting. But
it's also got me realizing I maybe have anthropomorphized chimp behavior of the human, or the expectation, because I feel like I watch my kids way more than they pay any attention to the fact that I'm in the room. But maybe I'm wrong. Maybe they're constantly watching me.
Well, we talk about kids being a sponge, and there's the old saying, little pitchers have big ears, there are all sorts of anecdotes about kids paying attention and learning things and knowing things that adults really didn't think the kids were paying attention to. Yeah. So maybe this, kids learn from models, from parents. And so, but additionally, maybe the kids are also more motivated by the little apple reward that they were going to get than the parents were.
Could just be it.
Yeah. So speaking about the difference between ages, kids to adults, that's development. But once you become fully developed, then it's all about aging. And you know what? According to University of Arizona researchers who just published a paper in the Proceedings of the National Academy of Sciences, there's nothing we can do. All this aging research, all this stuff we keep reporting on, intermittent fasting, just starving yourself. Oh, exercise, all that. I'm going to try and stay young. Doesn't matter. You're going to age. and you're going to die. Aging is a fact of life.
But can we slow it down so I can live to be over 200 like I planned?
Yeah, so I think that is the question there, because what they did is they put together a mathematical equation, and they modeled aging in a multicellular organism like humans on the computers. And so this model they put together takes into account that there are a couple of factors at play. And so you basically have the competition in balance between cells that need to be eliminated because they're not performing well. And so those are the cells that are, as they become more decrepit, their metabolism stops working as well. Those are the aging cells. And so if we could just get rid of all of those cells, then maybe we would be just fine, right? But then on the other hand, you have cells that take advantage of this and become cancerous and grow out of control and go to the other extreme. So the balance point within multicellular organisms has to be keeping the whole thing balanced. So allowing sluggish cells to accumulate at some rate, or allowing cancer cells to proliferate. And they say if you do one, you can't do the other. You can't do both at the same time. So they say this equation expresses that aging is an incontrovertible truth and an intrinsic property of being multicellular. One of the researchers, Zazzle, says it's no surprise that we're all going to die. Lots of things are obvious because they're so familiar to us. But really, why is it that we age? And this study starts to get at that and starts to explain why we have aging in the first place. Once you get past development and you end up in a point of bodily maintenance, it's a balance between getting rid of or allowing sluggish cells to accumulate or allowing these cancer cells to start going out of control. And so this starts answering this question. And the researcher says, why hasn't natural selection stopped aging yet? She says implicit in that idea is the idea that such a thing as non-aging is possible. So why haven't we evolved it? We're saying it's not just a question of evolution not doing it. It can't be done by natural selection or by anything else.
Well, it would make sense that it can't be done by natural selection because the name of the game in natural selection is reproduction. And if you've passed reproductive age,
right,
then there isn't a huge benefit to you staying alive in terms of natural selection.
Right. I mean, we've got things that we talk about like the grandmother effect and other possibilities, and men tend to be fertile for many, many more years than women. But yeah, reproduction does have a huge role to play there for sure. So there are interesting questions about it. And yeah, aging does seem to really hit the fast track after reproductive age has passed. But they're not looking at the mechanisms per se. They're looking at this as a mathematical demonstration of why you can't fix both problems at the same time. So we're either going to we can fix part of it and maybe slow it down a bit. Maybe we can fix the cancer and figure out how to stop cancerous cells from taking advantage of the system. Maybe science still has something it can do. But, like you asked in the beginning, Blair, maybe it's not so much a matter of stopping aging as it is slowing it down. Mm-hmm. And there's the difference between aging and then senescence. And some people undergo a very long senescence, and that's really like when the body's really breaking down. Mm-hmm. So if we can shorten the period of senescence, so it's just a much shorter period and we age gracefully, maybe that'll be better. Maybe you can still live to 120 or 180. Maybe that's possible.
200.
200. Okay.
Don't cut her short, Kiki.
I'm aiming for 200.
Sorry. Yeah, maybe the answer lies in the naked mole rat.
Perhaps.
Perhaps they don't get cancer.
Right.
Why not?
So you're gonna live to 200, Blair, as a naked mole rat.
It
This
could be
is
worse.
how to do it.
Could be worse.
Too much fun. But anyway, according to math, we're all gonna die.
Yay! We won't
Math.
overpopulate the earth.
Yeah.
Hooray!
This is This Week in Science. Justin, what you got there?
So we know all life is going to eventually come to an end for all of us, mathematically speaking, except for Blair.
Right.
But the origin story of life on Earth is still a bit of a mystery. The primordial soup, the molecular muck, the deep sea thermal vent event, whatever it was, we still don't know. There is a prevailing hypothesis known as the RNA world hypothesis that earlier forms of life may have relied solely on RNA to store genetic information and to catalyze chemical reactions. According to a new hypothesis, life later evolved to use DNA and proteins due to RNA's relative instability and poor catalytic properties, not because of just RNA being that good at transmorgifying things, but because of a partnership between nucleic acids and small proteins called peptides. So this is sort of it sounds like it's jumping ahead a little bit. It's like, what? We've got these tiny proteins, but I thought RNA was supposed to lead the protein. So how could there be proteins that are already, according to two new papers from biochemists and biologists at the University of North Carolina, Chapel Hill, and University of Auckland, their peptide RNA hypothesis contradicts the widely held RNA one, or RNA world hypothesis.
Interesting.
Yeah, so before there was life on Earth, there were some simple chemicals. Research this centuries previously revealed the chemicals for the soup that created the basic building blocks of life already were there. So we got building blocks, but now how do we get building? How do we get single cells? How do we get proteins to single cells, to animals, plants, life on Earth? So the mystery is how amino acid building blocks were first assembled according to a coded nucleic acid templates into the proteins that formed the machinery of these cells. So the prevailing theory, RNA world, posts that RNA that is still involved in coding and regulating and expressing genes today somehow elevated itself from this world of amino acids and chemicals to give rise to the first peptides. Like they somehow they created the first peptides. Carter and Wilson argues that RNA could not kickstart this process alone, but because it lacks a property they are calling reflexivity. It cannot enforce the rules by which it is made. RNA needs peptides to form that reflexive feedback loop necessary to lead to eventual life forms. So at the heart of the peptide RNA theory are enzymes so ancient and important to life that the remnants are still found in all living cells, even in some subcellular structures, including mitochondria and viruses. There are 20 of these ancient enzymes called amino-acyl tRNA syntheses, which I'm just going to refer to from now on as ancient enzymes. The special attributes of the ancestral version of these enzyme superfamilies and the self-reinforcing feedback system they would have formed with the first genes and proteins would have kick-started early biology, driven the first life forms to that complex mitigation of, or creation or proliferation of all this diversity that we see on the planet. This is co-authors Peter Willis, PhD professor, physics at the University of Auckland. He says, compared to the RNA world hypothesis, what we've outlined is simply a much more probable scenario for the origin of life. We hope that our data and the theory we've outlined in these papers will stimulate discussion and further research on questions relevant to the origins of life. Ancient enzymes, there's 20 amino acids. Each one of them recognizes 20 amino acids that serve as the building blocks of proteins. So it's a sort of a little bit of a chicken and egg kind of a thing, right? You have this RNA that can code and say, here's what happens next, but it can't actually put the building blocks together. That needs protein to be present. But how do you get to the protein? So these 20 ancient enzymes belong to 2 structurally distinct families, each with 10 ancient enzymes in those families. Carter's recent experimental studies showed that the 2 small enzyme ancestors of these 2 families were encoded by opposite complementary strands of the same gene. Made one gene, just one bit of coding, and it's creating either this or that, right? It's creating 2 families. The simplicity of this arrangement, with I love the way they phrase it here, with its initial binary code of just 2 kinds of amino acids, suggests it occurred at the very dawn of biology. Moreover, the tight yin-yang interdependence of these 2 related but highly distinct enzymes would have stabilized early biology in a way that made inevitable the orderly diversification of life that followed. These interdependent peptides and the nucleic acids encoding them would have been able to assist each other's molecular self-organization despite the continual random distributions that beset all the molecular processes in the beginning, Carter says. We believe that this is what gave rise to peptide RNA world early in the Earth's history.
That's fascinating. So instead of starting off the starting block with RNA, it was peptides, which is and then the peptides would have given rise to RNA eventually after time and mutations and change. But how fascinating that these so instead of the RNA hypothesis, maybe it's the peptide RNA
hypothesis. So
peptides to RNA, and then viruses.
Yeah, they're also pointing out that, and I think likely how they got to this is not being to overcome some of the obstacles in the RNA hypothesis. They're pointing out that they've shown it's impossible obstacles would have blocked any transition from pure RNA world to a protein RNA world to an onward towards life. Such a rise from RNA to cell-based life would have required an out-of-the-blue appearance of an ancient enzyme-like protein that worked even better than its adapted RNA counterpart. This is an extremely unlikely event that would have needed to happen not just once, but multiple times. Once for every amino acid in the existing gene protein code. It doesn't make sense, he says. And then goes on to say that the 2 scientists are fully aware that RNA world hypothesis still dominates the origin of life research field. Quotey voice, that theory is so alluring and expedient that most people just don't think there's any alternative, Carter says. But we are very confident there is.
Yeah, and this is there's another interesting study that was out this week also suggesting that that these RNA and I guess in this case maybe it was the peptides, but that the phosphorus that's involved in the amino acids that builds these molecules, that it all could have gotten its start not in clay like we've talked about before, but actually just in little droplets of water. And that little droplets of water can actually have formed the labs, the little laboratories where components came together and mixed in a water solution. Yeah. I don't know. We keep looking around at space. There are building blocks all over the place. Maybe wherever we find water, if these things come together at these microscopic scales and combine and recombine, and if you have something like these peptidases, these peptide enzymes, they can do things. They can catalyze chemical reactions that end up making what they're doing valuable and interesting and build up to the RNA.
People say life didn't just happen. It kind of did.
Yeah.
But over a very long time after, a bunch of steps.
Yeah, but it did just kind of happen.
It did just kind of happen. And it could have happened all over the place.
Yeah, and the simpler solution to it happening that is all, it keeps looking like we're finding a much simpler chain of events. Yeah, exactly, Kiki. The more likely it's gonna be in lots of places.
Yeah. Well, I want to hear more. You've got another story about. We're going to link this to the alien life story, but that'll come in the second half of the show, right? You teased that you've got an alien life story. So talking about life starting and just happening. I'm going to tease it again, everybody. We're going to talk about alien life in the second half of the show. So you can just think on that.
Have we discovered it? Question mark. Is it here on Earth? Question mark. That and more, but in the second half. Because right now it's time for a different segment.
Yeah, because right now, you know what time it is? It's time for Blair's Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels.
What you got, Blair?
I'm so excited to talk about the unknown that we were just talking about. The great unknown out in space. There's a lot of that, but there's also a lot of great unknown here on Earth, particularly in our oceans, which is the thing that I like to rhapsodize about often. There's a lot of things we still have to learn from the ocean, and the Animal Corner is full of two pretty fascinating discoveries this week in marine life. This first one is about nudibranchs. Nudibranchs are very colorful, beautiful sea slugs. And a particular group of nudibranchs off of the coast of Sicily were being studied by Dr. Trevor Willis, senior lecturer and course leader at University of Portsmouth. And they saw something kind of interesting. They knew that nudibranchs eat hydroid colonies. So hydroids are a stage of life for a lot of animals called hydrozoids in class Hydrozoa. They're closely related to jellies and corals, but they look almost more like plants, but they're actually a kind of animal. And there's not a lot of nutrients in them. If you think about a jelly, you think about a jellyfish, it's kind of just a water bag. There's not a lot of food there. There's not a lot of nutrients there. So they wanted to look at these nudibranchs and really if they really are just eating hydroids. They were especially interested because they thought that if you did really just subsist on hydroids, they would have to eat so much of it to get what they need that they would actually deplete hydroid populations. So the risk would be that their food would run out before they could actually reproduce. So that's a pretty terrible life history strategy. Well, what they found that these nudibranchs were eating polyps that had just eaten. So over half of the nudibranchs' diet was actually made up of not the hydroids they just ate, but the zooplankton that the hydroids just ate.
Double your pleasure, double your fun.
I was actually thinking about something like a turducken in a weird way, right? But so
this is an aquatic lower trophic level turducken.
Yeah, an
Yeah.
invertebrate turducken. So over half of the food was actually the food of their food. So they're doubling their attack rate on prey that has gorged on zooplankton when compared to hungry counterparts. They're not bothering with the empty hydroids.
So it's kind of like when I have a hamburger and salad.
No.
My food and the food my food eats?
It's like if you ate a cow right after it ate grass,
Oh.
and when we looked at your poop, it was full of grass.
Okay.
So not exactly. So they're actually using this species, the hydroids, as a fishing rod, quote unquote, to get access to the zooplankton, which normally they wouldn't be able to get. Nudibranchs, little sea slugs, they're not the best filter feeders. They really need to, they have a radula, a little scraping tongue, so they really just have to eat stuff that they can nom on. And so they're calling this new strategy kleptopredation. Kleptopredation. Yeah, the way they figured all this out, they looked at the nitrogen stable isotope levels in the nudibranchs, the hydroid polyps, and the zooplankton. They found that the nudibranchs had lower levels of the nitrogen stable isotopes than they would expect feeding only on polyps. So they were filling their tummy up with something else, and that's where they found all of this out. This actually is, even though it sounds pretty cruel, a win-win for the hydroids and the nudibranchs, because remember before I was saying if they're eating these little polyps all the way up, they can deplete populations. Well, the hydroid is usually kind of this big branching structure, and so one quote unquote animal could have all these different polyps. And so actually by grabbing the polyps that just grabbed food and having to eat less, they're extending the life of these colonies. So it's a weird win-win, but it's definitely a win-win. Now, the reason they had to come up with a new name for this, kleptopredation, is because it's actually a combination of something they did know about called kleptoparasitism, which is when one species takes food killed by another, like hyenas grabbing a lion's fresh kill.
Aha. Right.
And direct predation, just eating another animal. So then they put those together, kleptopredation. Dr. Willis says that our ability to understand and predict ecosystems in the face of environmental change is impeded by a lack of understanding of trophic lineages. In scientist speak, that basically means we can't save a space if we don't know who's eating who. And he also says there's a lot to learn from research. I love this quote. While we have some great results, like any science worth its salt, it raises more questions than it answers.
Absolutely.
And I love that. I think I might have to write that down somewhere because we talk about that on the show all the time. What's next? What are they going to study next as a result of this discovery? So they found this whole new feeding strategy. Chances are it's not just these one nudibranchs off the coast of Sicily. There's other animals doing this. So I'm fascinated. I can't wait to hear who else is a kleptopredator.
Who else is kleptopredating? Yeah, the idea that they are eating them so soon after that the hydroids have eaten, that there's not time for digestion to have taken place. You don't lose the energy to metabolism, which is what, if you eat the cow who ate the grass, by the time we get the cow, that grass has already been metabolized into muscle mass and
we only get a tenth of that. We get a tenth.
Exactly. And so we get a tenth of it. And so if you're faster and you eat the food that has just eaten, then you're getting all of the food. You get to have the benefit of all of it. And as they said, if it's helping their populations survive, then that's a good strategy for them.
Yeah, absolutely. Everybody wins, even though it kind of sounds like the hydroid loses a little bit.
The hydroid definitely loses, and so does the hydroid's prey.
Yeah,
but
I got eaten twice today. Says the zooplankton. Okay, so
yeah.
The other story I brought tonight is about another animal that exhibited something we weren't really expecting. And that's our oyster friends, little oysters. Oysters are extremely important animals, before I go any further, for a bunch of reasons, but mainly why?
They're delicious.
No.
Oh, sorry.
They're filter feeders.
They're filter feeders. Yes,
Yeah.
of course. That's why they clean our
So-
water.
Too many oysters, water gets too clear, and habitats collapse. Not enough oysters, water gets too dark, habitats collapse. Filter feeders are extremely important to the delicate balance of a stable ecosystem in the ocean. So now that I've explained why we actually care, besides the fact that they're delicious, they exhibited something very interesting in a laboratory experiment from University of Bordeaux, France. And they wanted to see if oysters could hear. So invertebrates in general, jury's out on their hearing. We definitely don't see, actual hearing structures, so it's hard for us to identify if they're just feeling vibrations, if they can actually hear, if they respond to stimuli of sound. So this is a study looking at that. And and specifically they wanted to see if they could hear things that are similar to noise pollution in the ocean, which I brought up recently on the show talking about noise pollution. We've talked about noise pollution and light pollution in the ocean pretty recently. So it's a really important part of when we think about how we are disrupting ocean ecosystems. It's not just the oil that we dropped in the ocean. It's stuff as simple as an idling zone nearby, or a motorboat or a tanker. So all those things actually have different frequencies of sound. And so each of them may or may not affect marine animals, and if they do, it's going to be in different ways, potentially. They looked at 32 oysters in a laboratory using a loudspeaker to play sounds underwater in a range of frequencies. And previous experiments looking at oysters have found that there's actually a behavioral response when oysters are stressed. So when they get threat or stress senses, they will snap shut. And so they wanted to see what frequencies of sound, if any, cause the oysters to close their shells. They found that when acoustic energies were high enough, oysters rapidly closed their shells at sound frequencies between 10 and 1,000 hertz. Their maximum sensitivities at low frequencies were between 10 and 200 hertz. So that was the space where they were the most responsive, was between 10 and 200 hertz. They found that the idea here is that oysters can hear tidal cues, maybe, and that's why they need to be able to do this, is that they can hear tidal cues that trigger behavior that helps them respond to rising tides. So if they hear rushing, crashing sounds like on your white noise machine while you're going to bed, you're listening to your waves.
Yeah,
That's
how would they
sound.
know?
That's a
Yeah.
sound that the oyster could hear getting louder and go, oh, better shut tight.
And you say hear, but maybe, hearing is really just that transduction of vibrations. And
They
so
are
if they're
sensing.
feeling, they're sensing vibrations.
They
Yeah.
sense the sound. Most marine noise pollution is due to cargo boats, and cargo boats, for the most part, are at low frequencies within this 10 and 200 hertz. Other things in that 10 to 200 hertz range: explosions, seismic research, pile driving, and wind turbines. Things that are at the higher frequencies that are less likely to bother oysters: small recreational boats, jet skis, water bikes. They're too high to see a response, at least in a lab.
Great. So we can
oysters.
still just ride our water maggots around as much as we want.
Sure.
Yeah.
Well, it more tells us that what we need to focus on are these cargo boats, which, Elon, hurry up and make the electric cargo boats. We need them now. Those cargo boats are so loud, and it has a huge impact on marine ecosystems. And if it's affecting everything all the way down to little oysters, we have some things to be concerned about because, as I said, they're a really important part of the ocean ecosystem.
Yeah, and so the question is, how do they respond to these noises? How long do they stay closed after a particular vibratory assault? So if it's a car, and like you said, the cargo boats are loud. So whereas one kind of watercraft or activity, like a wind turbine, the wind turbine might be in the right noise range, but maybe it's farther away and not as loud. So maybe that sound of the wind turbines doesn't propagate as far. So and then the cargo ships, because they're louder, maybe they propagate further. Maybe they have a larger area of impact.
Right. Absolutely.
And then based on the loudness, how long do these little filter feeders stay closed?
Right. So maybe then we can take life history information we have about the oysters, like the time of day when they are doing the most filtering, and reduce sea traffic at that time. There's lots of things that we can do to try to take care of this, because if the oysters are essentially sleeping at night, then maybe cargo ships should be moving in and out then. But then, of course, there are potentially other species that are being impacted at that time. So you have to figure out, it's a big part of it, right, is figuring out when we will have the least impact, because things as we do them now have an impact on the natural environment. That is locked in. There's no way around that. But we just want to make sure that we don't turn around and find that our filter feeders aren't doing their job and an ecosystem collapses. So it's a part of the equation, pH, ocean pH is a part of the oyster equation. There's all sorts of things that can stress out oysters. We've just found another one of them that we can keep in mind as we move forward.
I guess my question is, I'd like to know when and where oysters sleep.
Yeah, well, that's a whole nother question, right? And then how do you define sleep? Right? But definitely there are times of day where filter feeders like oysters and mussels are more active, and days where they're and times where they're less active. So
yeah, we know there's the oyster season. During the winter they're less active. During the spring and summer they're more active. So at what point there are different seasons when the oysters are fresh and feedable. But yeah.
But there are also times of day too that oysters are more productive. So
right, and they're going to open or close as the tides go in and out, and so there are many factors that probably regulate their behavior. That vibrations are one of them.
Yep.
Yay! No.
Save the oysters.
Save the oysters. And so this is how we all become a little bit of a part of the blue oyster cult. No.
Cowbell?
Cowbell? Do we have cowbell? We've come to the end of the Animal Corner and the first half of our show. This is This Week in Science. It's time for us to take a quick break for a few words. Please do stay tuned, because we've got alien life and electrical brain stimulation coming up, among other wonderful stories in the second half. So stay tuned, everybody. We will be right back with more This Week in Science after this. Hey, everybody. Thank you so much for listening. This is This Week in Science. And do you know what's available right now? Oh, yes, you do. Yes, you do. If you head over to TWIS.org, you will be able to click on the wonderful image that allows you to pre-order the TWIS 2018 Blair's Animal Corner calendar. You can get that now. We have not yet ordered the calendars because we're in pre-order stage at this point, but Blair has completed the calendar. We have many, many images, and if you are watching the show right now, Blair is giving you a preview of many of the wonderful hand-drawn images that she has created for this next year's calendar. And this calendar is a coloring calendar, so you will have the great joy of getting to color in and interact with this beautiful calendar month after month. It'll make it a much more personal calendar experience this year. Some beautiful pictures here that Blair has created. Blair, this is looking so much fun. I hope that everybody out there heads over to TWIS.org to pre-order this amazing calendar, which not only has the coloring pictures for you, but also special holidays and science days that are of interest to people who listen to TWIS. So you never miss a cool holiday coming up in 2018, or an episode of TWIS for that matter. Additionally, while you're over there at TWIS.org, you can, if you're interested in perusing the other things we have to offer, because it is November and that holiday season of gifting is coming up very soon, you can buy someone you love a mammoth lumbar pillow. Blair drew the art for that and many other items that are available in our Zazzle store. So click on that Zazzle link at the TWIS.org page, and you'll be able to find all sorts of goodies for the person you know and love who enjoys TWIS t-shirts, mugs, mouse pads, hats, all sorts of fun things. Or you can even buy stamps that you can affix to your holiday cards, which is a fun thing also. So you can head over to the Zazzle store, click the Zazzle store link, and that will take you there to be able to see all those wonderful fun things. Also, while you're at TWIS.org, if you are interested in supporting TWIS directly through a donation, you can click on the donate button, which will take you to a PayPal interface where you can choose your amount, and it's a nice, easy PayPal interface. Click on that donate button if you want to donate to TWIS. The other option, if you'd like to be a part of the supporting community for TWIS, is to click on our Patreon link. That's on the header bar of TWIS.org, and that will take you to our Patreon page, patreon.com/thisweekinscience, where you can click on the link to become a patron and choose the amount that you would like to support us at for each episode or over the course of a month. And there are special fun things that we do for you supporting us at various levels of support on Patreon. We also send you little notes every once in a while. And when we do have videos and episodes and special things that come out, you get to have first look at those items very, very often. That's Patreon. And for those of you who are not really into the whole money thing, you know, the in-kind, the being-kind donation of helping us to increase our reach, to get out to more people, that is a very important thing. Marketing costs a bundle. But if you can just tell one person this week about TWIS and get them listening to it, that's going to help so much. So you can share us on Facebook, on Twitter, on Instagram. Take a picture of yourself listening. Where do you listen to Twitter? I mean, not to Twitter. Where do you listen to TWIS? Where do you listen? I'd love to see pictures on Instagram. Hashtag TWIS, right? Hashtag This Week in Science. Share it on Instagram. Share it with your friends. Let people know. Let them know that you're listening all the time. I'd like to know too. And for those of you who are interested, also, we are going to be in Denver, Colorado this Sunday through Tuesday, and we are going to be at the Entomological Society annual meeting in Denver, Colorado. And if you're at the meeting, I hope that we see you there at our live show. But if you're not at the meeting, I hope that we will be able to see you at our meetup. I don't know where it's going to be yet, but we will be announcing it on social media as soon as we have a time and a location. So keep your eyes peeled. Everyone out there, thank you so much for your support. We really could not do this without you.
Is the dollars and cents all these authors receive? If miracle wonders were held in their looks, why waste precious time and try selling their books? Why sit and wait for your publishing royalty. If one has real power, who needs reader loyalty? If you could travel by thought to a mystical place, why go to book signings and fight for shelf space? Why would you wait for your agent to call?
And we're back with more This Week in Science.
Yes, we are. Justin, what you got there?
I do have a story about alien life and discovery of what it would look like, but that's going to be later in the show. First, over 100 years ago, in a theory of general relativity, Einstein predicted that gravity does not act instantaneously as Newton had thought, but instead propagates at the speed of light. Cody voice of Neil Cornish, physicist at Montana State University. The speed of gravity, like the speed of light, is one of the fundamental constants of the universe. Until the advent of gravitational wave astronomy, we had no way to directly measure the speed of gravity. But over the recent shows, physicists have made some really big progress in this direction. And it looks like they've got the first bounding, this is air quotations, bounding of the speed of gravity because of these observations. Initially, first LIGO detectors, detection of gravity waves, constrain the speed of gravity to within 50% of the speed of light. That does not mean that it is half the speed of light. This is very important. Bounding it within 50% of the speed of light means that they've got a boundary of slower than the speed of light and faster than the speed of light. Much faster.
Right.
Because so far, this is where that's 50%. And then paper published last week in Physical Review Letters, Cornish and his co-authors came up with a new constraint that bounds it to within 45% of the speed of light. Again, that's not even slower than the last one. That's still above the speed of light and below the speed of light, but a narrower bounding, okay, a narrower range where they can say, aha, we've got a solid detect. This is a speed of light. So this is, they are right now using all the different gravitational waves emitted by the binary neutron star merger, along with some light detection. This was a very good event because it's not just gravity. There's also a light event that you can kind of detect and say, oh, that's part of this speed of light. This is part of it, right? They've got it to -3, 10 to the 15 power, and 7 times 10 to 16 times, 7 times 10 to the 16th power times the speed of light. So it's a lot more than the speed of light than it is below the speed of light. But again, this is just the current range of which we were able to make these detections. It does not mean that gravity is faster than the speed of light or slower than it. It just means we haven't got the actual radar gun on it yet, right? One of the things they're saying is that if we have more, part of what helped them do this is we had LIGO detectors that were located 1,800 miles apart. And they're kind of what they're saying is that they had 5 of these gravitational wave events to compare using 4 detectors in different places on the Earth. They believe that's the good radar gun that they need to have ready, or the amount of data that they need to have come in, and then they could get it down to maybe 1% of the speed of light. So we've got our first sort of ranges where it can't be below this or over that. And while that might sound like there's a lot of work to be done, it actually excludes a number of alternatives to general relativity when it comes to gravity.
Yeah, so this is the really interesting aspect of this.
Right. So scalar tensor out. Theory by the Horava-Lifshitz, gone. Gravity and biometric gravity, they're just gone.
Yeah, they're the modified, the MOND. These are certain modified gravity theories that they've been using to try and explain how everything works in the universe. And the MOND theories and MOG theories, they modified of gravity, they actually, many of them don't take dark matter into account. So this is also at a certain point maybe going to be mixing in whether or not there is dark matter.
Right, because by playing with these constraints on the speed of gravity, you can have different explanations for the expansion of the universe. You may have worked out an idea that doesn't include dark matter, and so it doesn't need to exist. So by ruling out a lot of these alternatives, by putting this first constraint on, it's already progress into a correct model, right? It's already tremendous progress. So this is only going to be a story that continues to unravel, and it's going to be fun to follow. But right now, 45% within the speed of light, mostly above, though. That's really what's fun too.
Yeah, well, no, I mean, where they are now, they're getting really, I mean, the light, the gamma ray light and the gravitational signal and the gravitational wave detector LIGO, it was a matter of seconds between the arrivals that they measured. So, I mean, this 45%, 45%, nope, no. I mean, basically what we're down to is light and gravity travel at the same speed, because that 1%, those few seconds there at the very, very edge of detection, that's error, because we're talking about the 100 million light years that the light and the gravity had to travel across. Maybe something slowed one of them down a little bit.
Or more likely, it's just our detectors. Or
it's our detectors. Exactly.
The things that are going to slow those down aren't going to make so much of a difference. I don't think. I think that it's just our ability to like I said, run a proper radar gun at it, have a proper detector to detect these speeds is really difficult.
And now gravity, we've almost got the exact speed, but pretty much it's the speed of light.
It's still, speed of light, at least at this point, is definitely not ruled out.
Yeah, it's not more. It's gravity does not. We did not see gravity travel faster than the speed of light. I just want to make that clear.
7 times 10 to the 16th times the speed of light. I mean, it's
the plus of the minus. Exactly. Yes.
That's a lot of over still.
But it's not.
But it is. It is quite a bit of over. That's quite a bit of over.
Yeah, because it's boundaries, but we saw it. Boundaries. It's not going to go faster than the speed of light. I'm just saying that now. That's my prediction. That's just error bar.
I will jump right in there with you on this one.
Just whiskers. You're saying it's just the whiskers on the graph?
It's just the whiskers.
Box and whiskers.
Yeah. And the probability of the overshoot is really, really slim. I'm gonna say, everybody out there. But I think this is all very exciting for not just narrowing down the speed of gravity, but also narrowing down the potential theories that explain how everything works.
Light travels at the speed of gravity. Wait, what?
Yeah.
I haven't seen you in gravity years. My goodness.
That's right. Change our cultural references. That would be fun. Cultural references. You know what cultural references show up in our brains, right? When people say words, our brains have responses to those words. And we can use functional magnetic resonance imaging to actually be able to observe changes in activation of the brain in response to different words that make you feel different ways, that make you think different things. So a group of researchers decided they wanted to use MRI readouts to identify, this is researchers from Carnegie Mellon University publishing in Nature Human Behavior. They wanted to use this fMRI, functional magnetic resonance imaging, to see if they could create a learning algorithm, train a computer to identify suicidal brains. And they did.
What?
It worked very effectively. However, we're also dealing with a very small number of subjects that were involved. And there are certain caveats to the research that the researchers themselves bring up at the end, which is that this was a pretty long measuring test, and most people, it'd be hard to have them sit or lie in an fMRI machine for a half an hour or more to do this kind of test without some kind of noise or errors taking place. But they were able to train a computer algorithm based on images of brains from suicidal and control individuals that were shown words related to suicide and not related to suicide. So they were showing words like death and distressed and other words as well. And they identified 5 regions of the brain and 6 words that helped to identify brain patterns of behavior related to suicidal thoughts. And then they trained their algorithm. Out of the 35 participants, the algorithm correctly identified 15 of the 17 suicidal patients and 16 of the 17 control group people. And then they took the suicidal thought group and they split them up into a smaller group. Those that had attempted suicide and those that had not. And a different algorithm was also able to categorize those patients 16 out of 17 times. So they say that there are particular brain responses just to the words, but those brain responses also trigger emotional responses. And in the brains of the suicidal patients, or even those that had already attempted suicide, very often words related to suicidal thoughts triggered emotions related to shame. And there was a different emotional response than was brought up in the control brains.
Wow.
So, not that we're going to go around fMRIing depressed people all the time, but this could potentially lead to a treatment at some point in the future to actually determine if people, if someone says they're suicidal, you take them seriously.
Right.
But many people never say they are.
Mm-hmm.
And so maybe there will be some kind of diagnostic test that will go around it. And maybe they can come up with a treatment, or not a treatment, but a test that doesn't take a really long time to do, that maybe it's a very quick test. Maybe there's even something that they can map to EEG, or a very simple diagnostic that doesn't take so much effort. Yeah. So anyway.
That's fantastic.
Yeah, AI for identifying suicidal brains.
Yeah.
Yes. And there is a difference between a suicidal brain and someone who's just having had some suicidal thoughts. It's a very complex issue. Actually. Justin.
Uh-oh, what'd I do?
You know what else is complex? Life in the universe. I want to know about it.
Okay. All right. Let me get my cursor back.
Why don't you want to tell me this story?
Okay. So we know that there isn't a single answer in human history or in science that can be answered with, it's aliens. Yes, you can actually answer that way, but you are just as incorrect in answering anything and everything with, because of Toxoplasma gondii. Although T. gondii will, on odd occasion, actually be the correct answer. Still, let that not preclude us from discussing aliens.
Right.
Because there are at least 100 billion planets in our galaxy alone. At least 20% of them are likely to fall into habitable zones. And even if 0.001% of them can figure out how to use those basic building blocks that are probably everywhere in every one of these systems, those planets involved life. That would mean that there would be somewhere in the neighborhood of 200 1,000 life-harboring planets in our galaxy, and we know that there are billions and billions and billions of galaxies beyond our own. So this is a study published in International Journal Astrobiology. Scientists from the University of Oxford show for the first time evolutionary theory can be used to support alien predictions and better understand their behavior. They show that aliens are potentially shaped by the same processes that shaped you and I and all life on this planet. Natural selection isn't a thing that we came up with, right? We didn't like invent it. It's not like genetic code per se for natural selection. These natural selection is largely how life forms play out on a macro scale.
Yeah, that there are environmental forces. There are differences. If this is a life form, then it's passing on traits to further generations. There are certain standards to natural selection and the way that it works.
Yeah.
Yeah. Heritability.
So this is, I'm going to lose my place here for a second. Where is it? This is titled Darwin's Aliens. Is the title of this study, if anybody wants to look it up. It's actually kind of a fun read, right? So going for the Sam Levin, a researcher in Oxford's Department of Zoology, says a fundamental task for astrobiologists is thinking about what extraterrestrial life might be like. But making predictions about aliens is hard. We only have one example, not of aliens, but of life, that is life on Earth, to extrapolate from. Past approaches in the field of astrobiology have been largely mechanistic, taking what we see on Earth and what we know about chemistry, geology, and physics to make predictions about aliens. In our paper, we offer an alternative approach, which is to use evolutionary theory to make predictions that are independent of Earth's details. This is a useful approach because theoretical predictions will apply to aliens, even if they're silicon-based and do not have DNA and breathe nitrogen, for example. So using the idea of an alien natural selection as a framework, the team addressed extraterrestrial evolution and how complexity will arise in space. Species complexity has increased on Earth as a result of handful of events known as major transitions. These transitions occur when a group of separate organisms evolve into a higher level organism. When cells become multicellular organisms, an example, both theory and empirical data suggest that extreme conditions are required for major transitions. And one of the things you could also think of, not just as a single cell that becomes multicellular, but when that multicellular organism starts having single-celled organisms living in its gut, there's all of this in place. Paper also makes specific predictions about the biological makeup of complex aliens, offers a degree of insight into what they might look like. I love one of the photos that they got, or the sketches that they have. It looks like a tardigrade, like the tardigrade of all tardigrades, right?
Yeah, a multi-limbed tardigrade. Yes,
Right.
it does.
Oh, yuck. Yeah,
No,
but okay, so I read this article. I definitely saw it. It got shared around quite a bit already, and I feel like, once again, kind of got this sensationalist title to the article that made it sound like Star Trek was right. Everyone's just humans walking around with a slightly different skin color or a funny wrinkly forehead. So that's not what it's saying.
No.
What it's saying is that they anticipate the same pressures to quote unquote shape or put processes into place that will influence these other life forms for the same reasons.
Right.
What I would like to know, though, is what is new about this paper other than some new fun pictures? Because honestly, anyone who has read sci-fi and even hard sci-fi that has alien life that is evolved on a distant planet, or that talks about humans spreading to different parts of the universe and evolving into different forms. I mean, this idea of natural selection acting on humans spreading elsewhere in the universe or on other alien species based on the planets where they come from. This is not new. We have talked about this before.
Yes, but we haven't put it into a paper out of Oxford by a zoologist. We've just talked about it.
No, that's my question though.
Maybe we should have written a scientific paper then. Jeez.
So, to Kiki's point, did they run some statistical analyses? Did they do anything kind of experimental in their exploration of this topic?
Read the paper. So yes, they did. They did a little bit of that.
No, that's why you're here, Justin. You're reporting on it. Tell me.
I know. So no, they just made some cool sketches and were like, wouldn't this be a cool looking alien? Let's write a paper about this.
So they run statistical analyses based on the assumption that natural selection will put pressure on these other rudimentary organisms on other planets, and
yes, that's it.
And yes,
and found
and they found that
yes,
natural selection.
You're not taking the bait here. I'm trying.
Yeah. So.
What did they find? What did they find that was new? What is it that the statistical analysis told them?
What they did was they came up with a couple of constraints or a couple of things that are going to drive. Which it's not that they're just taking it from the planet that we know to an extent, but they're saying it doesn't have to be the same biological makeup. But there's going to be what they're predicting is. There will be hierarchy of entities which all cooperate to produce an alien. This is sort of like humans with our microbes and all that sort of thing. Each level of an organism will have mechanisms to avoid conflict, that maintain or engage in cooperation, that keep the overall organism functioning. So, and sort of one of the things which I don't think is talked about very often, they're looking at the whole biome of an entity versus does it have 2 legs or 6, right? And instead of just focusing on how does gravity affect the length of a limb or something of this nature, there sort of are more building a discussion about the entire
more subtle forces.
They're looking at an integrated approach for sure.
Yeah. But they did not find aliens in outer space.
No, nor did they have a rendering of a space lady doing a fan dance or something like that.
No.
Actually, that did come up too. That was also possible.
Can you do the fandango?
Yeah.
No. Yeah. So no aliens in outer space. Maybe there are aliens here on Earth with us, but we don't know that. They didn't find those either. But natural selection, universal, just like gravity and the speed of light. Constants. Oh, you know what else is going to affect us in space as we spread to the far reaches of the universe? Space brain. Unless, of course, we can figure out how to stop it. Researchers from the Medical University of South Carolina were funded by NASA to look into the question of what happens during spaceflight to astronauts' brain structure. And so this is another MRI type study where, based on studies here on Earth where they had people volunteer, and I don't understand that people would do this, but people volunteered to be bedridden for 90 days and keep their head in a particular orientation to approximate the effects of microgravity on the head and the brain and the body. During that study, they did find effects on the brain, and what they saw, and they saw problems related to the visual system. Researchers in that study, they saw the brains and muscular responses of these patients and the neuroplasticity that occurred. That neuroplasticity in the brain occurred during bed rest, but there was a crowding occurrence at the top of the brain. So things like there's openings, these sulci, these openings up at the top that kind of give space between the folds and the gyri of the brain. That space went away, and the brain kind of started squishing up to the top of the skull.
Wow, that feels like a slow motion concussion, or sounds like a slow motion concussion taking place.
Right?
That's weird.
So there's this shifting going on, and she's like, all right, this is happening here on Earth, which this could just be a weird effect of the study. But let's look at the brains of astronauts who have actually gone to space. And there's been a symptom that's been reported among astronauts. It's called visual impairment intracranial pressure syndrome, or VIIP, VEEP for short. And they think, we've talked about like the eye pressure, the interocular pressure that affects the visual system. The astronauts come back, their eyesight is impaired, kind of blurry. But we don't know exactly what's going on there. And they think it's a redistribution of cranial fluid or body fluid, even that the spinal pressure, the fluid is redistributing. But they looked at these astronauts' brains, and they did actually find the same effect as what they saw in the individuals who had been on bed rest. And they saw these openings, these sulci at the top of the brain, go away. And so there was a change, an actual change to the structure of the brain. So this intracranial pressure is a thing. The brain is either swelling or fluid is moving the brain around. There is something actually structurally changing within the brain during microgravitational experiences that doesn't happen here on Earth, and that affects the experiences of the astronauts. And this could be part of the overarching problem of a lot of brain issues related to being in space over long periods. So, should we go to space? What does that mean for our brains? Should we become spacefaring? Should we send people to space for longer periods than we currently have, the longest being actually over a year now? What'll happen?
I guess we need gravity helmets if we're going to go.
Right.
Because artificial gravity, that's totally something that's legit possible, right?
Yeah. Well, if we're in something, a situation like an intrasolar system trip where we're under thrust at a certain level of thrust that could approximate gravity, if you can cause enough vehicular thrust, spacecraft thrust through space to give yourself gravity, then your body feels gravity.
Sure.
And that would be okay.
So we can go to Mars. We just have to go really fast.
We just have to go really fast. Yeah.
So these people that were bedridden for 90 days, did their brain go back to normal?
I believe so. That's a really good question. I'm not exactly sure what the after effects of the being bedridden were, but it is true that astronauts who go to space for long periods of time actually do experience trouble reacclimating to being on Earth, that they do have balance issues. They have vision problems. They
come back taller.
Right. They come back a little taller, but they also come back weaker. And so there are lots of issues. They also have nausea issues. There are all sorts of problems that people don't really talk about for the returning astronaut, especially those that have been at the International Space Station for extended periods of time.
Oh, which, by the way, the Google Maps or Google Earth, I think it's just the Google Maps. You can zoom out now, and you can go to Mars and Venus and the moon and do like a map, like zoom in and out of portions of that. But they also have the International Space Station as a little like walkthrough. You know how like you can sometimes go down to street level and walk into like a restaurant that's taking photographs all around so you can kind of see what the environment is like inside. They have that up for the International Space Station now. I encourage everybody to go take a look at it. But I can sort of also picture from having wandered around in it for a while how odd it would be to just sort of be a largely two-dimensional creature again, where there's going to be something, you know, in front, behind, and left and right of you that you can interact with. Whereas if you look around the walls, there is no walls. There's the ceiling and the floor could be workstations, right? Like everywhere you look, there's stuff there that you could be interacting with and doing something with. So that I could totally understand how disorienting it would sort of be to be back in a two-dimensional world where there wasn't a laptop on the ceiling that you could go and work on if the room got crowded. Like all of a sudden you're back in it, all new parameters, of course, with the gravities there pulling on you constantly when it wasn't before. Yeah, I mean, it's a very different environment. It's a very unintuitive environment. And so just another layer of the trouble with spaceflight. But I do encourage everyone who isn't listening to this show to go ahead and go. I want everybody who listens to this, stay here with me. Don't leave me. I'm not going.
Right now, we reported a while back. So the 90-day bed study that I mentioned, that one occurred early 2000s. There is a new NASA-funded bed study. It's a bed rest study. It's under NASA ENVIHAB, ENVIHAB, and this mission is a 70-day bed rest study. The purpose of this study, it's also called the VAPOR study, VAPER, is to study the effects of pressure on the astronauts' eyes and optic nerves in space. But 12 volunteers will spend 30 days in bed with a head down tilt and in a 0.5% carbon dioxide, mimicking the space environment thought to cause vision problems in astronauts.
I have so many questions.
My only question is, am I still allowed to like watch TV or play video games while?
Yes, so it's interesting. You can, but you're not allowed to change your orientation. And so you actually must stay in a bed on your back with your feet slightly elevated with respect to your head or your head below your feet.
Oh my God. And you probably can't get up to go to the bathroom.
Nope.
It's all you're there. See,
but you have an opportunity. Hopefully it comes with access to like a Netflix thing set up. You get to binge watch everything on Netflix, even things that you've passed over because you won't be interested in them. So there's a little bit of a benefit to get. I've watched everything on iTunes University. I've gone through all of the books on tape that I always wanted to read, but never had the time.
Yeah.
Because at some point your vision probably starts to go. So books aren't going to do it. You need an audio.
You got to be listening to things. Maybe learn a language or something like that. But this study, I guess, got underway as of October 2nd, and it'll be interesting to see in the next couple of months as the study is completed and they've got their measures, what comes out of that ENVIHAB NASA bed study. So let's keep an eye on that for when the news comes out, you guys. And everyone who's listening, if you see links, send us updates if we don't see them. Let's move to the end of the show and get these last stories out of here.
It's quick news time. Pew, pew.
You know what's, this is the quick news segment of the show. And you know what's the fastest thing that humans have ever controlled?
Gravity. Light.
No, an X-ray laser pulse.
Oh.
Huh?
Yes. So microscopic world of atoms and electrons. It's really tiny and these things move really quickly. When there's a chemical interaction, the parts of the molecules, the electrons that are changing places, they're hard to keep up with. So if we want to make little movies of chemical reactions, we need a light source to record those reactions with that is as fast, if not faster, than the reactions themselves. And so researchers at ETH Zurich have shortened the pulse of an X-ray laser down to 43 attoseconds, the shortest controlled event ever created by humankind. And to get an idea of how tiny 43 attoseconds is, which is this is at about the level of time that these electron transfers take place at. Get this. It is scaled up. There are as many attoseconds in 1 second
Mhm.
as there are seconds in 31.7 billion years.
No.
So what
No,
you're
you
saying
kept
is
going, and I was like, oh, nope, she's going to stop. Nope. 3, not 3, 30, 30, okay, 31 months?
7
No, years, no,
billion
billion years.
Yes. As many attoseconds in 1 second as there are seconds in 31.7 billion years. And so this X-ray laser pulse is 43 attoseconds. This is about 1,000 times shorter than the LINAC in LINAC coherent light source. And additionally, this is shorter than a University of Central Florida team that had a 67 attosecond pulse. 43 attoseconds. We're going to make movies of chemical reactions. We're going to see how these things work. Maybe, and like if you want to know what kind of movies would be really interesting, how about photosynthesis? How do electrons transfer through the chlorophyll during photosynthesis? And can we use that information to make better solar systems? The answer is probably yes. And so they're trying to make even shorter attosecond pulses even now. And then I said early in the show that I had an electrical stimulation for the brain. Well, not really, because what I have is evidence of a particular kind of electrical brain stimulation not working. Researchers and people with epilepsy there have been lots of claims about a particular kind of brain stimulation called transcranial alternating current stimulation, TACS. They said it could help memory-related brain waves and stimulate memory. But in fact, now published in Nature Communications, direct stimulation of the brain with TACS, transcranial alternating current stimulation, found absolutely no effect on brain waves associated with memory. So if you're interested in stimulating your brain with electricity for memory, maybe TACS is not the thing that you want to be using. There's not much evidence there. However, this does not suggest that transcranial direct current, TDCS, doesn't work. There's evidence that that does work and other things as well. So anyway, TACS, not good, not necessarily a memory booster for your brain. So maybe don't do it. Yeah.
So you're saying don't shock myself in the brain with electricity.
Well, unless maybe it's direct current.
Oh,
alternating current.
that's what I've been doing wrong.
You're not getting
it's
it.
the
Right.
whole.
And maybe just
so
use magnets.
finally, there's one place where Tesla's invention isn't as good.
That's right, Tesla. Not necessarily good for
Tesla
memory in the
versus
brain.
all over again.
Yeah. All right, tell me about T-Rex arms. Let's get this done.
Yes, T-Rex arms. You know, why don't you ask a T-Rex to paint your house? Why doesn't a T-Rex like to wear a party hat? All these questions come with answers.
You can never take it off.
They have tiny arms. So the T-Rex, they're about 40 feet long. They're about 20 feet tall. They have strong, long, powerful tails. They have about 5 foot long skull, and they have hands. They have little arms that are less than a meter long, less than 3 feet long. They're pretty short. A lot of paleontologists think that they are vestigial. They have absolutely no purpose. They're just silly. But paleontologist Stephen Stanley of the University of Hawaii at Manoa said on October 23rd that he thinks the limbs are well adapted for vicious slashing at close quarters. Not sure how you would get close enough to get slashed at with a 5-foot head on top of a 20-foot body, but that is his hypothesis.
I'm just imagining the whole slashing. It's like
Yeah.
weird baby slaps. I don't.
Yes. So they had longer arms. The T-Rex ancestors had longer arms used for grasping, but at some point they got smaller and smaller, probably when their head was getting so much bigger, so that they didn't tip over.
The mouth.
Yeah. So their head got so big and heavy that they had to lose weight elsewhere, or they would no longer be able to be bipedal. So that's the kind of main hypothesis, is that the hands just shrank over time so that he didn't fall over on his face.
Yeah, a little counterbalancing, right? Using the arms for a little
Stanley
balance.
says that based on the bones that we have found of T-Rex, those arms are very strong. Those bones are robust. They could sustain impact of slashing. So why would you have big, robust bones in these vestigial arms? It doesn't exactly make sense. Each arm also has 2 sharp claws about 10 centimeters long. 10 centimeters long, that's like 4 or 5 inches. So that's a pretty long claw to be doing nothing. On top of that, they had 2 claws instead of 3, as most dinosaurs do. So by having 2, you actually have heavier pressure. And the edges of the claws are beveled like a bear. So they're not flat like grasping claws. All of that makes him say perhaps they're slashing claws. Other scientists are definitely not convinced. They say, like Thomas Holtz of the University of Maryland says, although strong, the arm of the fully grown T-Rex would barely reach past its chest, greatly reducing its potential strike zone. But here's the other side. Perhaps because the T-Rex arms grow more slowly than their body, young T-Rexes, young baby T-Rexes, would have proportionally longer arms, which means potentially juveniles could use them for slashing prey.
Interesting. Right. So then you'd have a change of strategy over time, maybe, but also maybe just help yourself get up when you fall down.
No, couldn't do it.
But no, for the juvenile. For the juvenile to be able to. And also, you know,
the juvenile could protect themselves a little bit better before its head was fully formed, possibly.
Right. And there has been talk that the T-Rex was very much a scavenger, perhaps more than a predator or a primary predator. They were more likely stole kills or found dead or you know, found an exhausted, maybe just chased down once in a while. But for the juvenile, and I don't know, this is the thing we need to know, is whether or not they traveled like a family. Like, is this a pack of wolves? Is it a pack of T-Rexes together? Because if the juveniles are out on their own hunting, they may be less likely to scare off other prey, other predators, so they may be not as good at scavenging. So maybe they actually needed to do a bit of hunting early on, and that's why, you know, and then, you know, those arms would have been really necessary for that stage of life, but then became less necessary as I could just scare off any other predators after they made their kill and just take it from them.
Oh, yes.
You know what I'm going to take from you?
Kleptoparasitism. What?
Yeah.
Salt. I'm going to take all the salt. I want all the salt.
All right. Well, many Americans, including Kiki, apparently consume more salt than they really need. Now, in a study appearing in the Journal of Agriculture and Food Chemistry, scientists report that people who can easily taste salt have differing amounts of certain proteins in their saliva than those who are less sensitive. The finding could help explain why some of us have a hard time shaking the salt habit and could potentially lead to the development of actually edible low sodium foods. Consuming too much sodium increases risk of heart disease and stroke. These are leading causes of people dying in the United States. Lower sodium products are on the market, but people don't buy them because they don't taste good. To develop more palatable products, researchers are trying to gain better understanding of how the body processes and perceives saltiness. So saliva has been thought to play a role. Unclear, though, exactly what it is about spit that makes the difference in how people have salt perception. So a small study was done. Thomas Hoffman and colleagues sought to fill in the knowledge gap. Researchers classified volunteers into sensitive and non-sensitive groups according to how salty the participants thought various sodium chloride solutions were. Using liquid chromatography and mass spectrometry, team identified several salivary proteins that differed between those who could readily detect and couldn't quite tell if something was salty yet. Surprisingly, they found the largest differences, not just in separate from that, they found big differences in resting saliva compared to saliva produced after swishing around. The resting saliva sensitive subjects had higher amounts of endopeptase enzymes that cut up proteins than the non-sensitive subjects. So it's this higher level of enzymes in their spit as they're swishing stuff around, and those who could taste salt right away than those who couldn't. Researchers suggest enzymes could be modifying sodium channels, which would increase the amount of sodium that gets into cells. Also, alternatively, and perhaps contrary to their first idea, the enzymes could be cutting proteins into in the saliva to produce salt-enhancing peptides in people who are sensitive. So they don't know exactly why, but it could be one of those two things. So then they could create, if they could somehow get this enzyme already in low salty foods, they could create low salt foods that people who were not as sensitive but liked salty snacks would enjoy them. And then the people who were salt sensitive would be like, oh, I can't eat salt ever again because everything is even the low sodium salt chips are too salty now. And then they'd have to come up with a product that had no salt in it at all, I guess.
Or you can just eat spicy food, according to a study that was published this week in the Journal of the American Heart Association called Hypertension. Researchers discovered that it appears that salt and spiciness activate brain activity in overlapping parts of the brain. So eating spicy food can actually enhance your sensitivity to salt. And they found that people with a high preference for spicy foods have lower blood pressure numbers and consume less salt in general.
Wow.
So potentially you can just eat spicy foods and not eat as much salt. So spices, some spices can help you not have so much.
Spices are the salt of life.
Yeah, and so that, those spice mixes that are the salt replacements, there's a little bit of science to back them up. Yeah, but this is us backing up to the end of the show. Everybody, I want you to remember, we might not have salt on our website, but at TWIS.org, you can pre-order the Blair's Animal Corner calendars. My cat is attacking an object from underneath the door that's making all sorts of noises. I don't know if you're hearing about that. Little hands poking through. Additionally, Denver, Colorado, if you're going to Entomology 2017, we hope we see you there. And if not, maybe Monday or Sunday nights, we'll have a meetup and we will be able to meet you there in Denver, Colorado. We will put information on social media. Additionally, fun stuff coming in. You know, Blair, how you asked for a whole bunch of limericks?
I sure did.
Well, we got a wonderful email from Fernando Haro.
Yes.
And he said, and we have another limerick that is very specific to what has science done for me lately that's going to come up in a couple of weeks on the show. But these are not specifically the what has science done lately version. But Fernando says, I was just listening to my TWIS backlog, episode 640, while working on some schoolwork when I heard Blair's request for some limericks. Now, I'm not the best at poetry, as English is not my first language, but I still gave it my best shot. I hope you enjoy this silly compilation from yours truly. This is for you, Blair. Dr. Kiki and Justin and Blair always try to be courteous and fair, with countless new stories to cater your worries. Global warming deniers beware. Justin starts the podcast with an offer so that maybe you don't have to suffer. The disclaimer's in place for all the human race. A researcher is never a bluffer. Dr. Kirsten will be glad to share how their listeners are always aware about the tidbits of science that keep our lives in balance, whether it's hobbies or work or healthcare. You might find all the news fascinating. Those theories are indeed stimulating, yet the hosts beg to differ, their opinions quite stiffer because 5 as a sample is frustrating. It might shock your cerebral cortex how can species become so complex. Allow your mind to now wander down in the animal corner where there's a ton of invertebrate sex. And finally, this should be your podcast of choice. Week in and week out, we're the voice. In all those darkest moments, science permeates our comments. TWIS saves us from the media noise.
Well done. Well
Thank
done,
you, Fernando. Thank you for those wonderful limericks. Thank you for that fun, science-based, twist-based poetry moment.
More.
And now you've just made Blair greedy for more. So everyone out there, do you have limericks for Blair? More limericks. And if you can theme them up to what has science done for you lately, bonus points, +1. So it's now time for us to end this show. Thank you, everyone, for listening or watching. Thank you to our chat room for being here, making comments while we did the show. Everyone's applauding Fernando right now and his limericks. Also, thank you to Identity4, Fada, and Brandon for helping out with aspects of the show that we find difficult to do ourselves and keeping the show going that way. Thank you so much. And now I would like to say thank you to our Patreon sponsors. Thank you to A Honey Moss, Aaron Luthen, Adam Mishkan, Alec Doty, Alex Wilson, Andy Groh, Arlene Moss, Artyom, Ben Rothig, Bill Kersey, Bob Calder, Braxton Howard, Brendan Minish, Brian Hedrick, Brian Condren, Brian Hone, Bruce Cordell, Byron Lee, Charlene Henry, Christopher Dreyer, Christopher Rappin, Colombo Ahmed, Craig Porter, Dale Bryant, Dana Pearson, Dale Garcia, Daniel Garcia, Darwin Hannon, Daryl, Dave Neighbor, Dave Wilkinson, David Friedel, David Simmerly, David Wiley, Donald Trump, The Dubious, Dougal Campbell, E.O., Edward Dyer, Emma Grenier, Eric Knapp, Eric Wolf, Felix Alvarez, Flying Out, Gary S., Gerald Sorrells, G. Burton Lattimore, Gerald Onyago, Greg Guthman, Greg Riley, Haroon Sarang, Hexator, Howard Land, Howard Tan, Aluma Lama, Jacqueline Boyster, Jake Jones, James Dobson, James Randall, Jason Dozier, Jason Olds, Jason Roberts, Jason Schneiderman, Jim Tellier, Jim Drapeau, Joe Wheeler, John Atwood, John Crocker, John Gridley, John Ratnaswamy, Keith Corsell, Ken Hayes, Kevin Parachan, Kevin Railsback, Ksenia Vokova, Kurt Larson, Larry Garcia, Layla Louis Smith, Mark Mazzaros, Marjorie Mark, Marshall Clark, Matt Sutter, Matthew Litwin, Mitch Neves, more Cowbell, Mountain Sloth, Nathan Greco, Orly Radio, Patrick Cohn, Paul Staten, Paul Disney, Phil Nadeau, Philip Shane, Randy Mazuka, Richard Hendricks, Richard Onemus, Richard Porter, Rick Ramus, Robert Aston, Rodney, Rudy Garcia, Salgood Sam, Shuwada, Sir Frankadelic, Stefan Insom, Steve DeBell, Steve Lessman, Steve Mashinsky, The Harden Family, Todd Northcutt, Tony Steele, Tyler Harrison, Tyrone Fogg, Tainer84, and Ulysses Adkins. Thank you for all of your support. Yeah. Thank you for all of your support on Patreon. And if you would like to support us on Patreon, you can go to twis.org and click our Patreon link, or go directly to patreon.com/thisweekinscience. Remember, we don't need the Patreon. You can help us out simply by telling your friends about TWIS. That will help because it'll bring more people who might want to help us with Patreon. It works that way. On next week's show, hey, we may be broadcasting live from Denver, Colorado on Monday at 12:15 p.m. Mountain Time. We may be.
If the Wi-Fi gods permit.
If the Wi-Fi gods permit, that is true. But we will be back for certain next Wednesday at 8 p.m. Pacific Time, where we will be broadcasting live online. And you can join us and watch at twis.org/live. But if you can't make it, don't worry. Things get archived, and you can find all of our past episodes at twis.org/YouTube or at facebook.com/thisweekinscience or just twis.org.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory. Or if you have a mobile type device, you can look for TWIS, the number 4, Droid app in the Android marketplace, or simply This Week in Science and anything Apple Marketplacey.
For more information on anything you've heard here today, show notes will be available on our website. That's at www.twis.org. That's TWIS.org, where you can also make comments and start conversations with the hosts and other listeners.
Yes, or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twisminion@gmail.com, Blair at blairbaz@twis.org. Just be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter where we are at TWISscience, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a limerick that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
It's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just better understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma. Got the eye. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you learned anything from the words that we said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
Okay, everyone, we have come to the end of the show. It's time for the post-show, and I guess there's a lot of post-show to go. Kind of. Blair said that we had lots of things to talk about tonight.
We have some items to discuss.
We have business items coming up. Justin's not back.
We'll start with Adam.
Yeah, we can have this. We can have conversations with Adam. Calendar. Your calendar is looking amazing. Looks
Thank
good.
you.
So you think we're ready for ordering?
Yeah, I just need to go over what I think my judgment on the whatever weird sciencey and animal-y holidays are going to go on there. You know, like you're welcome to look and put in your 2 cents, but I think that's okay. I tried not to overload it. I mainly just wanted to decide which like calendary holidays we want to include, because I've done it a little bit different both years to this point.
Mhm.
I've tried to do all of the like school and work related holidays and a couple of fun ones, but otherwise leave a lot of religion based holidays out of it since that's very denominational. But this time I kind of just threw everything on there. So
Mhm.
right now what I have is, be ready?
Yes.
New Year's Day,
Great.
MLK Jr. Day,
Great.
Groundhog Day, Lincoln's birthday.
Wait, hold on. Let me go to my calendar.
Okay.
Hold on 1 second here.
Okay.
January. Let's go to January. All right. New Year's. I want to look at this.
Mhm.
All right.
New Year's Day, Martin Luther King Jr. Day, Groundhog Day.
When is that? Yeah, great. Okay. Cool.
Okay, Lincoln's birthday,
Which is in
February
February?
12th.
Great.
Yes.
Okay. That's also Darwin's birthday.
Yes, so that's also Darwin Day.
Okay, great. Cool.
Yes.
Okay.
I did not include Ash Wednesday.
That's
It's
fine
like
with
not a
me.
holiday. Okay, Valentine's Day I included.
'Cause that's fun.
On this calendar, they call it Chinese New Year, not Lunar New Year. Do you have a preference, or do you think? What do you think?
What do people call it? Is it Lunar New Year? Is that what they call it?
I don't know. I mean, they also called Indigenous Peoples Day Columbus Day.
Columbus Day.
So I
Yeah.
deselected that and I manually added in Indigenous Peoples Day.
Good. I think we should change that one for
Yeah.
sure. That's great.
Um,
Let's see. Hold on. Lunar New Year, right?
yeah,
Friday, February 16th.
correct. Should I change it
Yes.
to say Lunar New Year?
It also says Chinese New Year. Hold on. Lunar New Year. What does it mean?
Mhm.
Is the first day of a secular, sacred, or other year whose months are kind of
Right. Like, are there other Eastern religions that consider that the New Year, and by calling it Chinese New Year, we're being like non-inclusive?
right.
This
That's
is
a great
the question I'm
That
asking.
is the question. Yeah.
Maybe I will. I'm gonna deselect Chinese New Year.
Spring Festival.
add
Chinese New Year, also
in-
known as the Spring Festival in modern China, important Chinese festival celebrated at the turn of the traditional lunisolar Chinese calendar.
But is that just how that calendar is based then, and not a
Right.
specific
Hold on.
like?
Yeah.
Then it would just be Chinese New Year because they picked a lunar cycle to call it a New Year.
Huh?
But if it's actually based on a celestial event, then maybe we give it back to the moon.
Yeah.
So it's based on the moon, but it says Wikipedia. Thank you very much. Chinese New Year is celebrated in countries and territories with significant Chinese populations, including mainland China, Taiwan, Hong Kong, Macau, Singapore, Indonesia, Malaysia, Thailand, Vietnam, Cambodia, Mauritius, Australia, and the Philippines. It's considered a major holiday for the Chinese and has had influence on the Lunar New Year celebrations of its geographic neighbors.
Uh-huh.
Okay, I'm gonna call it Lunar New Year.
Yeah.
Okay. Great. I did not include Family Day. I don't know. I've never heard of that. I did include President's Day, Washington's birthday,
Great.
International Women's Day,
Great.
Daylight Savings begins, St. Patrick's Day, Spring begins. Do we want Palm Sunday or Good Friday on there?
I know what those are, so probably
No.
Okay, Passover.
Is that important to you?
Yeah.
Can we do this in vocal fry? I was promised vocal fry.
All right, fair enough. Okay, so Passover.
But we've never had Passover on before, have we?
No. So it's more a question of whether, for me, are we also including Easter? So I would say if one goes, both
Right.
goes.
Absolutely. This is
Okay.
we're going to de-religiousize the and focus on a science-based calendar.
So you're saying remove both.
yeah.
Yes.
Okay, great. This is why I wanted to ask. Cesar
Oh, wait,
Chavez
can we just get rid of-
Day.
Cesar
Can we get rid of
Chavez
Chavez?
Day.
That's
huh?
going to be on there. April Fool's Day.
That's fun.
What about Administrative Professionals Day?
What is
That's
that?
not one I had considered.
I've never. Well, they work that day anyway because
Yeah, they do.
they have to show up. So
Okay, it's gone.
No.
It's gone. Arbor Day, May Day, Cinco de Mayo.
Yeah.
Okay,
let's call it 5th of May Day.
no.
Oh.
Mother's Day. So we removed Passover and Easter, so I will also remove Ramadan. What is Victoria Day?
I have no idea.
Great. Memorial Day is in. Flag Day is in. Father's Day is in. Summer begins. What
It's
about
a Canadian holiday.
Okay.
Victoria Day is for our Canadian neighbors.
okay?
Did
Do
we
we want it in
hire a
there or
Canadian
not?
company to come up with the calendar?
What?
Is it a Canadian company that came up with this calendar?
Who knows?
We do have Canadian, but we can't have every holiday.
Okay, so it's gone. It's gone. What about
Sorry,
Canada Day?
Canada.
What about Canada Day? We can have that, I think. That's on July 1st.
Canada Bashing Day.
Independence Day, since that's on there, we'll do Canada Day. Labor Day. What about Grandparents' Day?
Oh, you can't take the grandparents out. I didn't even know it was a thing, but
No,
it's
I've
something
never,
that should
I've
be
never
celebrated.
done
I know.
Grandparents'
I
Day.
kind
But
of
now I
kind of feel
will
like
if it's on there.
It's a BS holiday, but we'll leave it on
in.
there. Okay. Fall begins. Canadian Thanksgiving.
Oh yeah, let's do that one.
No.
Okay, great. Okay.
Yes.
No.
Yeah, sure. Why not?
It's actually based on like being thankful as opposed to devastating
Eating
an
until you
entire.
hurt. Oh.
Yeah.
Yeah, they have a great track record.
Yeah,
Okay.
Canada's
I
better
removed
with
Columbus Day.
Thanksgiving.
Replace it with Indigenous People Days. Halloween is in.
Good.
Daylight savings ends. I don't know what Diwali is. Do you?
Diwali.
Never heard of it.
No, hold on one second. I have heard of it, but it's an Indian. Yeah. Oh, it's the Hindu Festival of Lights.
Oh, that's fun.
It is
It's gone.
fun.
Okay. I mean, if it's a religious holiday, we're throwing it out just like the other stuff, right? Okay. So Remembrance Day, Veterans Day,
I know,
Thanksgiving.
Lakshmi.
I will take Hanukkah off since we took all the other stuff off. And then winter begins. Christmas Eve and Christmas is not on there because instead we have Twistmas.
Christmas.
Do we want Boxing Day on there?
No.
Okay.
No.
And New Year's Eve. Okay, excellent. So then, do you want to hear the science
The ads?
holidays?
Yeah.
Okay, it's updating. I don't want to lose.
We changed for Hanukkah, we called it Twisika.
Oh yeah,
Called it
did I do that
Twisika.
this year?
Last year you called it Twisika
Let's see if I put that in there.
because it started on your birthday last year too, which was kind
Oh,
of
yeah.
fun.
I think I killed it. I think it's gone. I should do that though.
But then we'd have to go back and do Twisted On and Twist Over.
Do you still have Monkey Day?
Yes.
Okay, good. Because I like monkeys.
Good. Okay, so first of all, I have TWIS every single Wednesday for the whole year. Is that correct?
We should look at when it does it correspond to
Any
a
holidays?
holiday.
Okay.
Yeah.
So
It'll be close enough.
TWIS is on December 19th and December 26th. So
Great.
those should be fine.
They'll be fine.
So I
yeah.
put the year in review on December 26th.
The 26th, it should be fine, but yeah, that's the day after Christmas, huh?
Yeah. And it's
so
the
that'll
year
be
in review, so we won't need to do a whole bunch of research. We'll just have to go back and look at what we did before.
Yes.
So then besides that, I mean, TWIS is on my birthday, but I'll let that slide.
You sure?
Yeah. No, it's fine.
We can do TWIS without you for your birthday.
No.
You know,
Let's
it's every year we
see.
look at the calendar and we go, oh, yes, of course, we're going to do all these things. And it's like, wow, 52 weeks of TWIS, huh?
Yeah.
Mhm.
TWIS Giving is on November 21st.
But that's always Wednesday, and
before,
Thanksgiving is,
yeah.
it's always, so we just have to figure out, we just always
So
have to figure it out as it
also
comes.
guaranteed, we're not going to mess up TWISoween next year because it actually is on actual Halloween.
Oh,
Uh-oh.
and I
Is that a
can't do it.
problem? Okay,
Yeah.
so we're
That's
not
such
going to
a
have TWIS on Halloween then.
I have a child. We go trick-or-treating.
so do
we
we-
have to do a different day.
Do we want to adjust it or do we want to skip a week?
Mm.
Such a big decision this far out.
Well, we're making a calendar.
Just can it be
Mm.
TWISoween may not actually be a show? And in parentheses under it?
Well, it won't be, right?
Won't actually be a show. Stay tuned. Stay tuned for actual time. Something of that nature.
Well, I can't fit all that on there.
I know we can't put all those words in there. Do you think maybe for Halloween we could miss one episode?
Yeah, I think it's fine.
But
think
we
it's
still call it
absolutely
TWISoween.
Yeah, so I'm writing
TWISoween, because that's the holiday.
TWISoween,
No show.
no live show.
No live show.
That's good. That's it.
Okay, great. So we're taking
we
a week
can-
off.
And if we can say no live show, and as we come up to it,
Pre-record. Oh,
we can decide whether or not to change the night.
Yeah, we
it's
could
just-
also pre-record. That's true.
record.
Okay, so
But we
then
don't need to decide that now.
Right. Let's see, October 17th, October 10th, October 3rd. Okay. I think
If TWIS conflicts with the World Series, remove it. How about we just move the World Series?
Yeah, move
Come on.
I know I liked Kiki's comment in the thing. My alert went off on my phone and I was like, "Oh god, what's happening?" The way everything's been. I'm like, "Oh no, there's an emergency alert on my phone." No, it was the results of the World Series.
I'm not in San Francisco, and the Giants weren't in the World Series. So I was like, I didn't even know the World Series was happening.
Yeah, I didn't either until I went over to
Wow.
my parents' house and my dad was watching it. I was like, "Ah."
No clue.
So TWIS also falls on Dr. Kiki's birthday this year.
Oh, what day of the week is that?
That would be a Wednesday.
Wait,
then.
Wednesday. That would be a
Wednesday.
Wednesday.
Yeah. So August 1st is a TWIS day.
It's a Wednesday.
Yes.
Okay, so there will be a show. I
Right.
may take the night off.
Okay.
There
Great.
will be a show.
Excellent.
It's a Wednesday, and
Great.
it's a Wednesday, so maybe I won't take the night off. We'll see.
Right.
And yeah, we'll be here.
Okay.
Happy TWIS
July
Day
4th
to
is a
me.
TWIS day.
Happy TWIS Day to me.
Oh,
That's we
jeez.
will not be doing a show on July 4th.
Okay, so no TWIS on July 4th. Great.
Yeah, it's 4th of July. That'll be
No, that's fine.
It's a holiday day.
Totally
so,
fine.
great.
Okay. I'm trying to plan ahead. Okay,
I
let's
think that's
see.
great, so that we don't because we had a couple of times this year that we had told people we were going to do shows, and then something happened.
Yeah, I think we still always did them, but
Wait,
I think there were
yeah, no, I think
problems.
they all got
We were
done.
like, "Oh no."
Yeah.
I think we
Okay.
did them all. I can't remember what a show
had
that got
some-
No,
canceled
I
this
think
year.
we did them all, but I think there were a couple where we were like,
We moved
maybe
things
we shouldn't
around.
have.
Yeah, there were twist shoes.
TWIS shoes. Okay, let's see. I think I'm in April. Okay, I'm working backwards. I'm almost done. April 4th, March 28th.
And everybody
Okay.
who's watching right now, the people in the chat room know how this works. Some of you may be watching on YouTube if you're directly on the YouTubes. We go into business in the after show sometimes because we don't live in the same city. And so this
No.
makes it much easier to connect on things.
Some people like to know how the sausage is made.
And this is what we need to figure out for the calendar.
Okay. Also, wow, all the things are Wednesdays this year. TWIS lands on Valentine's Day. Yeah, I'm fine by it.
Yeah, I mean,
Justin?
I have a kid, and we usually don't go on dates anyway, so so Valentine's Day for me is we can do it on another night. I don't need I can
Yeah.
go out to dinner, and
Same.
I like
Justin?
and I love doing a
I'll
TWIS Valentine's
have to
show.
let you know much closer.
Okay.
This is not one of the not one of the decisions I am in charge of.
Right. I get it. Okay, great. So, that's it for TWISes. Okay, good.
Sweet.
I added in, let's see, we have National Bird Day. We have Appreciate a Dragon Day. We have
like
Penguin
that.
Awareness Day.
Good.
We have Squirrel Appreciation Day. International Zebra Day. Serpent Day. World Cancer Day. Darwin Day. World Radio Day. Galileo's birthday. National Hippo Day. World Pangolin Day. Nicholas Copernicus's birthday. International Polar Bear Day, National Pig Day, World Wildlife Day, World Congress on Nanotechnology, Pi
Cool.
Day, Save a Spider Day, National Panda Day, International Day of Happiness, World Water Day, Manatee Appreciation Day, National Ferret Day, World Rat Day, International Beaver Day, Zoo Lovers Day, National Library Workers Day, Yuri's
Good.
Night, National Dolphin Day, Bat Appreciation Day, International Cuckoo Day. It's one of my favorites I added. Earth Day, World Day for Laboratory Animals, World Penguin Day, World Tapir Day, Save the Frogs Day, Save the Rhino Day. May the 4th be with you. Wild Koala Day, National Teacher Day, National Shrimp Day, National Sea Monkey Day, Endangered Species Day, and International Museum Day, World Turtle Day, Don't Forget Your Towel, Rachel Carson's birthday, Slug Appreciation Day, World Oceans Day, National Black Cow Day, National Lobster Day, World Sea Turtle Day, World Giraffe Day, World Camel Day, Pink Flamingo Day, Save a Catfish Day, Tau Day, Nikola Tesla's birthday, Shark Awareness Day, National Zookeeper Week, Guinea Pig Appreciation Day, World Snake Day, Armstrong Moonwalk, Amelia Earhart Day, International Tiger Day, World Ranger Day, found out about that one, Dr. Kiki's birthday, of course, Owl Awareness Day, Sea Serpent Day. Did I say that one already?
You said Serpent Day before.
You said Serpent
Oh,
Day,
Serpent
but not
Day.
Sea
It's just
Serpent Day. Yeah.
Sea Serpent Day. National Sneak a Zucchini Day. This is a real thing. You're supposed to sneak a zucchini onto a neighbor's porch on August 8th of every year.
What?
No idea.
Yes. I loved it. Okay. World Cat Day, World Lion Day, the Perseid meteor shower.
Lion. Meteors. Yes.
Yes. World Lizard Day, National Honeybee Day, World Mosquito Day, Dog Appreciation Day, International Whale Shark Day, Species Requiem Day, another one of my favorites, Threatened Species Day, National Iguana Awareness Day, Bilby Day, International Red Panda Day, Plover Appreciation Day, Talk Like a Pirate Day, World Rhino Day, World Rabies Day, World Rivers Day. Those are all R's right in a row. International Raccoon Day, Butterfly and Hummingbird Day, World Virus Appreciation Day, World Teachers Day, Indigenous Peoples Day, Octopus Day, Ada Lovelace Day, National Fossil Day, I'm almost done, International Sloth Day, World Statistics Day, Wombat Day, Mole Day, National Mule Day, Jellyfish Day, World Philosophy Day, GIS Day, Fibonacci Day, World Anteater Day, International Cheetah Day, World Soil Day, Justin's birthday, Blair's birthday, Monkey Day, International Dalek Remembrance Day.
The Daleks?
Yeah.
What?
And Visit the Zoo
Remember
Day.
the Daleks.
So we
Okay.
got them.
On your list, is there a DNA Day?
No. When is that?
That's 425.
Of every year
Day
or of
commemorates-
2017?
It's 425 is a day that commemorates the Nature article that published details on the structure of DNA
DNA Day is April 25th of every year.
every year.
Yes.
These are the things that I have to figure out because I'm trying to make this easier for myself next year. So I can add things in differently as the same date of every year or as a specific date on a specific year. And some
Right.
things are like Red Panda Day is the third Saturday in April or something like that.
Right. They
It's
move
like,
around.
ugh. Okay, so I added DNA Day. What else do you need?
There's an Introduce a Girl to Engineering Day.
Oh, when's that?
That is 2/20. By the Society of Women Engineers.
February 20th?
Yep.
And that's February 20th of every year?
Probably.
No, probably.
And you got Darwin Day, I'm sure was on there.
Yeah. No. Is Introduce a Girl to Engineering Day every February 20th? Or you're not sure?
You look it up. I'm scanning a separate list here.
What's your list that you're looking at?
It's a list of, I'm having to scan past a bunch of them because it's a list of geek holidays.
Okay. I'm
so
gonna need your source material though, because some of the like geeky holiday sites are actually not accurate.
Mhm.
So just look up Kiki, or Kiki, if you could look up Introduce a.
I am looking it up right now, and hold on. Introduce a Girl to Engineering Day. Girl Day is an international event celebrated during Engineers Week each year.
Yeah, I'm not really finding an answer.
Mm-mm.
February 22nd.
Is that what you got? Okay, so you have a different day than mine.
And I don't, maybe
So,
it's
this
different
is-
locations. I think it just is a day that's celebrated during that week.
Oh, yeah.
Maybe different locations
shouldn't.
have different celebrations of it.
Yeah, I don't know. No, I think
11/23.
it's the 22nd.
11/23 is
22nd.
Fibonacci Day. You have Fibonacci Day?
Yeah.
I know that one's, you already did that? You have that already? Okay.
Yeah. Which is it?
11/23. 11/23 is
Yeah, I have it.
Fibonacci Day.
Yeah, I have it.
Because it's the sequence of that. Okay.
Yeah. Are we satisfied? Justin, are you still looking?
I'm still looking.
Okay.
I'm
this
gonna
calendar's
guess-
getting pretty.
Yeah, I'm going to
But
guess
I'll leave it.
Engineers Week changes every year. And
Yeah,
so that Girl
sounds
Day
like
would
it. So that's
change
a tough
every
one
year.
to add.
Yeah, I did not
We'll have
do
to
it as
find
a
out when it is.
recurring date.
Mm-mm.
We'd have to look it up for what it is this year. Yeah, that is a problem. Now I get what you're.
Yeah, because then this year I wasn't sure what was recurring and what wasn't, so I had to go through the entire list and check.
Yeah.
And so this year I was trying to be very careful to mark the ones that recur and the ones that do not.
Yeah.
So. Okay. Cool. All right. Well, then the calendar's ready to roll.
Nice.
Awesome.
So Kiki, when do you want to order this? Because I have to ask for a special coupon code and it only lasts like, what is it, 24 or 48 hours.
Right.
Do you want to wait till next week or do you want to do it this week?
Did we miss Mole Day? I missed Mole
No, I have
Day.
Mole Day.
Oh, but yeah. Okay, good. And Talk
Oh,
Like
yeah,
a
we
Pirate
missed
Day?
it. Yeah.
We missed it.
No, talk
Yeah.
about a pirate, like a pirate day. was
Talk
in there.
Talk Like a Pirate Day?
That was it.
Arr. You have to have Talk Like a Pirate Day.
Yeah, it's on there.
Okay.
For
Oh, this is
sure.
another one. Hobbit Day?
Oh, I'm not aware of that.
555 Revenge of the Sith. Is it 5/5?
That's silly. I'm just going through right now and making sure that there are any days with like 4 different holidays on them where it looks terrible on the calendar, but it's looking
Yeah.
okay.
September 22nd is Hobbit Day.
Okay, I'll see if we have room on September 22nd. It's looking good. Looking good.
But is that
yeah.
a Homo floresiensis hobbit or J.R. Tolkien.
No.
That's a Tolkien Hobbit, I'm sure.
Tolkien Hobbit.
Did you know that World Giraffe Day is always on the summer solstice?
I did not.
Do you want to know why?
I would love to know why.
Because it's the longest day of the year.
Oh my God, I love that. Scientists are funny. Geeks are funny.
It's pretty great.
Yeah.
Yeah, so do you want me to ask for the coupon code for like this week, or do you want to wait?
I think we could do it this week.
Okay, great. Do you know how many you're gonna purchase?
I need to figure out all those details. Why don't you give me a couple of days?
Okay, great. So I will get
can
you a
back to
coupon code. I will tell him that we need it on Friday. Does that sound good?
Let me get back to you.
Okay, that's fine. That is no problem. I will tell him that it's going to be this week, probably, just so he's like looking
Next
for
week,
my email.
or like next week.
Oh, next week. Okay.
Let's just say next week.
Okay, great.
Let's just say that. Yeah.
So in October, there's a couple days that are really full, but I don't really want to part with any of them. So October 3rd is Butterfly and Hummingbird Day. It is World Virus Appreciation Day, and we have TWIS. So it all fits, but the square is like full. And then October 8th is Indigenous Peoples Day, Canadian Thanksgiving, and Octopus Day. I think it's okay.
Yeah, it's okay. It's okay. People can
We'll
write
see.
over it.
We'll see. I in previous years I've tried to keep like one thing on a day, and this day I was just let's just do them all. Do all the things. So we'll see how people like it. Okay, so that's great. Cool. So we're all done. We're good.
Wow.
Woohoo!
Calendar complete. So that was the big thing. I will email. Do you still have the login to the I can send it to you if you don't.
Yeah.
To order the thing?
Probably in my email.
Okay. I
From
can
last
send
year,
it
probably.
to you. Yeah. Okay. So I will email this dude and tell him we're pretty much almost ready. And then for Denver, this is the other thing I had to ask. Do you need me to bring my laptop?
Yes.
Okay. I will do that. What are we wearing?
Whatever you want.
Okay. Do you want me to wear a twist shirt?
We can wear twist shirts for the show.
Okay.
We can, be there and represent TWIS, and then people we're wearing our shirt, and people will be like, "What's TWIS?" Maybe,
Great.
Because people want to know.
I want to know.
Right.
Will you tell me what is this orange twist word I see? I want to know. All right. Cool.
Right.
So I will do
Good
that.
calendar.
It's gonna be real cold. Justin, did you know that?
It's gonna be cold in Denver.
It's the lows are in the 30s and the highs are in the 50s.
Whoa.
Yeah. Yep.
Perfect. Love it.
So
That's not cold.
okay, cool. So I'm bringing my laptop. What else do I need to bring? Anything?
Hi, Kathy. I will call you or text you and let you know if there's something special you need to bring. I've been thinking, something you might want to bring in case you want to record interviews with people at the conference at some point. I don't know, Monday afternoon.
Okay, so I should bring my little microphone and attachment to my phone.
Yeah, I'm thinking I'm going to do something similar.
Cool.
Yeah. So we can do interviews maybe and trade off recording. I know yours works. I haven't tested mine yet. Yeah, I'm going to bring my iPad, and I'm also going to bring the Mevo. And we're going to see about being able to connect to the audio for the live recording. We'll figure it all out. Yeah. Justin, you bring a pen and some paper.
Yeah, maybe.
Okay, here's the other question. We're doing how long of a show?
90 minutes.
Okay.
And I have in the rundown, it's the date, 11/6/2017 in the rundown. I haven't set it up yet with the guests that we're going to have because I just got the names of those guests today.
Okay.
So we need to figure out those details. Who we're interviewing, what we're talking about. I just need to spend some time
Those are good details to have.
Yeah, good details we can figure out. But I'll get that. I'm thinking tomorrow I'm going to put that stuff in the rundown so you guys can think around. Let's see.
So how many stories do you want me to bring? How many buggy stories?
Yeah. So what did we do last year? We didn't bring that many stories.
Oh, in Baltimore?
In Baltimore, like 1 or 2 stories.
Baltimore or Philadelphia?
For the Baltimore.
Philadelphia, we had a lot of stories.
The Baltimore
live
show.
I felt like that one, the Baltimore live show, we had more stories available, but that we took up the time with the interviews is what happened. So we were over prepared, which is always a good place to be.
Right.
Yeah. What I'd like to do is theme it up because it's entomology. So
I don't know if I can do that, Kiki.
I don't know. I know it's so hard for you. So hard. But each of us find a bug-related story or 2. Because the interviews really are going to be the key thing. And I was thinking if we can get an extra microphone also, it might be neat to have like one audience question per interview. Also, like bring in somebody from the audience, which, kind of fills up the interviews. But, 20 minutes, if we do 20 minutes per interview, we have 3 interviews. That's an hour right there. So,
okay. So I'll bring like 2. Cool.
But I will also put in the names of these people and who they are, so that if we want to also theme up news stories related to their disciplines, like if you find a buggy story that's related to something that these people do.
Right.
Or we can do the opposite, which is find a buggy story that is unrelated to what these people do, so that we get more variety.
Might do a little column A, little column B. See
That's
how I feel.
Yeah,
because I found like 4 good bug stories this week that I plopped into that rundown just depending on whether or not I find more or not. I wanted to have some depending on how I felt. So cool. Very neat.
Yeah. So theme it up, entomology, bug stories. I'll get the stuff in the rundown tomorrow and we'll get buggy. We'll work out the bugs.
Excellent.
We'll work out the bugs. That's right. Yay, I get to see you guys in Denver. I'm so excited. Actually, so let's see. We can figure out one thing right now. Which is, let's see. So it's the, what is it? Denver Conference Center.
Where we're going?
The Colorado Convention Center.
Uh-huh.
That's where this conference is going to be. Let's take a look at that. The Colorado Convention Center. This is like right downtown. We're going to be, and I think our hotel is right there also, right near there. So we're near the Metropolitan State University of Denver. We're near the Civic Center. There's a few things there. Just outside this area, there's someplace called Great Divide Brewing. It's a cozy tap room for original craft beer.
Nice.
That might be a good spot.
Yeah.
There's a place called Ophelia's Electric Soap Box. Large space for dining and live music. View house, eatery, bar, and rooftop. I don't think I want to be on a rooftop when it's 50 degrees outside.
No.
Yeah. Oh, Denver Airport conspiracy theories. Thanks, Eric.
Oh yeah, I heard about that when I stopped
That's
by
awesome.
that one time.
That's right. Maybe, hopefully,
Underground
I should-
bunkers, yeah.
maybe I should just try and get Dave Friedel to recommend something. But
Yeah, that's probably a good idea.
does he ever go places? Does he do things?
I mean, one would assume.
I would assume. Do you know there's a line from The Tempest? Now I will believe there are unicorns.
Cute.
Thought is free. Oh, brave new world that has such people in it. I don't know if anybody has any recommendations for places to hang out in Denver. Good night, Fonzo Blessed. Good night, Fernando Lopez. Good night, people who are leaving. Yes, I understand. It's late. It's so late. Denver. I don't know Denver. I'm sure we could Yelp it, but I'd like to... I don't know places. The Secular Hub, is that a place, Strengths? Or are you just making that up?
Sounds like a place I would go.
I know. What is that? Oh, wait, what? Oh. It is in Denver.
The Secular Hub.
Yes, it
It's
is
hilarious.
in Denver to build and unite the community of secular organizations in the Front Range. They provide a meeting place for various meetups. But do they have a bar and a restaurant?
That's a good question.
That's interesting. The Secular Hub, secularhub.org. Strengths. What a interesting thing you've shown us here.
You know,
We definitely have fundraising dinners. Oh my goodness, how fascinating. People make interesting things. A local Wendy's. Yeah, thanks, Hot Rod. All right.
I like
will look
that
at 'em now.
Kiki is people make interesting things.
Right.
That's a good one. I liked it.
Thanks. Yeah. People do.
Wisdom.
This is my little bits of wisdom that pop out. Justin, you have any thoughts there?
Not yet. I'll sort it.
You'll sort it, huh?
When is this anyway? This is like weeks away, right?
It's Sunday. Everybody's gonna. Does everybody have their information for their flights?
Sunday.
I know when my flight is.
I know when my flight out is.
Yeah. That's all you need.
Okay, cool.
Just need to get there.
Yeah, I can do that.
It's the important part. Getting home is trivial.
Henry's Tavern. There we go. 4 stars.
I'm always like, give me all the delays that you can throw at me on the way home. That's fine. I just
Taco Bell.
don't delay me on the way to a location.
This is why we're going the day before.
Mm-hmm.
That we are not disturbed. We'll get there so that we get there on time. If nothing else, you'll be there in time. You might have to fly all night long.
How long is the flight to Denver?
It's like 2 hours.
Oh,
Is it?
easy.
Quick. Maybe an hour and a half. Yeah, it's quick.
Oh, I was thinking it was way long. I thought it was like 3.
I don't think so. Well, it might be the time change involved, so it might be
yeah.
longer.
Okay, let's see.
I don't think it's that
I'm
long.
looking it up.
That
2
mint.
hours, 28 minutes for me. On the way there. And on the way home, it's 2 hours, 53 minutes.
Okay, 3 hours then.
All right. I think I'm going to go to bed.
I'm
Okay.
tired as
But,
well, but I think I have some things. There are things on the map that might be good
Hmm.
for our meetup, possibly.
Cool.
Possibly.
Cool, man.
Yeah. I mean, we'll
It'd be
probably
cool
be
if they had like
4 of us,
private dining room.
right? Private. I
Run
don't need
down.
private. Don't need private. Just need a fun place.
Yeah, that's true.
Yeah. Yeah. All right, everybody. This is I'm not good at planning things like meetups and stuff because there are too many details and I just can't. I'm like, but what about this restaurant? And what about that place? And what about
That's
that?
why social media is so awesome. You
Yeah.
can be saying it as we're heading there.
Exactly. Where should we go, you guys? We want to have dinner Sunday night.
I like
Where
the
should we
brewery
meet?
idea. I'm always down for a brewery.
Yeah, there's a
You
couple
know, I love a
places.
sampler.
Yeah. Great Divide Brewing. It is, let's see. It's like 8 blocks.
Oh, that's stumbly distance.
Yeah, eight blocks or
From
so.
our hotel or
our
from the
ho-
convention center?
From our hotel.
Okay.
Maybe a little bit longer. 1, 2, 3, 4, 5, 6, 7, 8.
Also remember that in
I'm
altitude
finished.
you can't drink as much, Justin.
This is why I want a place with food.
Yeah.
You're the one going to the brewery.
I just like a sampler.
Yeah, which is why you need to remember it.
Nah,
Good night, Ed.
samplers are tame.
But at high altitude, Blair.
Eh, you don't
Alright,
tell
I do
me.
have to
I
go.
tell
I'm-
me.
I really just want places
I
to
am
eat
really
because
gonna
I
fall
like
asleep.
eating.
Halloween. My kids did not sleep at Halloween on account of they were eating candy all night. So
Mhm.
I'm gonna warn it. But they still managed to wake up extra early in anticipation of getting at more candy. So I got no sleep. So I'm gonna go. So
Okay.
All right.
say,
We'll go.
Say good night, Blair.
We're all going to go. Oh,
Good
my
night, Blair.
toe.
Say good night, Justin.
Good night, Justin.
Good night,
Kiki.
Kiki.
Good night. Good night, everyone.
How are we
Thank
getting
you for
worse at
joining
that
us.
with more practice?
I don't know. On my end, we were right in sync.
Oh, really? Wow. Maybe we got a little more lag than I think.
Thanks, everyone, for joining us for this show. We hope to see you again next week. Hopefully, we'll see you live on Monday afternoon. If not, Wednesday. Maybe we'll see you for a meetup. Stay tuned.